So the main topic of my presentation is going to be analysis of clinical trial with complex objectives. Here I'm going to concentrate on a fairly specific setup because my presentation is going to be built around a case study, a phase 3 clinical trial that employs multiple endpoints and multiple doses to better characterize the efficacy profile of this new treatment. But the general ideas, the general principles can be applied to a very broad class of multiplicity problems. And this broad class of multiplicity problems is characterized by the fact that there are multiple sources of multiplicity. For example, and this has been pointed out actually by several speakers today, you may have several endpoints, several dose placebo comparisons, evaluations of the treatment profile in several pre-specified subgroups, and so on and so forth. And in this presentation, I will talk about possible solutions, statistical methods for addressing those complicated multiplicity issues. And what I'm actually not going to talk about the specific methods as much as I'm going to emphasize the general principles. And for that reason, I'm not going to have any formulas in my slides. I hope you're not going to hold it against me. And the reason for that is actually, I think one or maybe several speakers pointed out earlier today that it's impossible to write a completely up-to-date guidance document. In fact, if you think about almost any scientific publication, as soon as it comes out, it's almost immediately outdated because in a couple of weeks another paper appears. Methods are constantly changed. Therefore, I would like us or myself in this presentation to really concentrate on the fundamentals, the underlying principles, things that would not be affected that much, when the next presentation on multiplicity or next publication on multiplicity comes out. So let me now provide a little bit of background for the topics I'm going to describe in this presentation. We'll assume that we have a clinical trial with tests, endpoint tests, or subgroup tests that are grouped into families. And here's the typical setup that you see on this slide. You have several boxes here. The first box includes the tests, the hypotheses of no effect that are primary. This is what makes or breaks this particular clinical trial. And then provided we've met those primary objectives, we can then move on to some other hypotheses, some other analyses, and those would provide additional evidence of efficacy, and those could potentially become the basis for additional claims. And therefore, when you look at family 3, even though I say here tertiary hypothesis, this does not mean that those are purely exploratory analysis. There's still, those analyses that could be translated into regulatory claims. And what's important for us when we talk about addressing those complex multiplicity issues is we need to find solutions that account for the hierarchical structure of this underlying problem. And to give you one example, this is an example that I'm going to use. I'm going to refer to multiple times throughout my presentation. This was a development program for lurazidone, that's the generic name of this product, in patients with schizophrenia. And the sponsor, this was a Japanese company, has actually conducted several clinical trials. This is just one of their phase 3 trials. And looking at the description of the main objectives of this clinical trial, you immediately see that there are several sources of multiplicity here. First of all, they had either 2 or 3 doses compared to a placebo in each of their phase 3 trials. That immediately gives rise to multiplicity. But on top of that, the sponsor wanted to characterize the efficacy of this new product by looking at the effect on several endpoints. The first endpoint was the PANSS total score. This is the endpoint that is used in almost all schizophrenia phase 3 clinical trials. And since the schizophrenia market is getting quite crowded, there will be a lot of competition when this new treatment enters the market. The sponsor felt, that they would need to have some important differentiating factors. It's not, and those came down finally to 2 additional endpoints. The first one is the clinical global impression, the severity score. And this is actually clinically somewhat independent of the PANSS total score. And secondly, and this is what you see at the bottom of the slide, they wanted to understand the treatment effect on the PANSS total score evaluated at a different time point. Instead of the 6-week time point, which is the regulatory expectation, they wanted to see how quickly the drug begins to work. If they do demonstrate that, this will be a very important differentiator for this new drug. And in fact, the reason they wanted to explore multiple doses in these clinical trials because obviously they wanted to improve success probability by having multiple shots on goal, and obviously that gave rise to multiplicity. And when it comes to multiple endpoints, as I explained a second ago, they wanted to have a stronger, what is the language, the terminology which is commonly used in the pharmaceutical industry. They wanted to enrich their product label by providing additional information that would be helpful for the prescribing physicians and patients. And in this particular case, they used a powerful gatekeeping procedure to deal with this complex multiplicity problem. And I was lucky to have contributed to this paper. And what you see at the bottom of the slide is the clinical publication that summarized the results of one of their phase 3 clinical trials. And the clinical trial publication actually emphasized the importance of having a pre-specified and powerful gatekeeping procedure that ultimately enabled the sponsor to get a very attractive label. When you look at their FDA label, you see that the results, significant results on those secondary endpoints are highlighted in the product label. And obviously lots of other examples. Now that I've joined Quintiles, we provide consulting services across the industry, and we constantly get requests to help sponsors deal with complex multiplicity problems. And I only have a couple of examples here. The list kind of goes on and on. And in this presentation, I'm going to look at a probably slightly more basic multiplicity problem. This is the case study around which this presentation is built. This was another lurasidone phase 3 clinical trial in which there were only 2 doses, which was 40 milligrams per day and 120 milligrams per day tested versus a placebo, and the efficacy profile was evaluated based on 2 pre-specified endpoints. The first one was the primary, and the second one was the key secondary endpoint. What do we do? How do we find solutions to multivariate, so to speak, in this case kind of bivariate multiplicity problems of this kind? I'm going to talk about general principles here, but I guess one of the first steps is to try to visualize the problem. And when we work with clients, with clinical trial sponsors, it actually sometimes takes a surprisingly long time to get to a simple diagram like this one. And this is a visual representation of this multiplicity problem. It's a 2x2 matrix. As you can see here, that when you look at the rows of the matrix, you get the tests for the primary and then for the secondary endpoint, and the columns correspond to those placebo comparisons. The general regulatory expectation back in the United States, where I came from, is that to be able to get certain claims, regulatory claims, into the product label, it is critical to control the global family-wise error rate in the strong sense. And there was actually a short discussion about kind of comparison of weak error rate control versus strong error rate control. I'm not going to spend too much time on this. I hope we'll have some time during the discussion, unless Armin objects. You're the boss here, Armin. Okay, but I will say just very briefly. I think the reason the strong error rate control is emphasized in the confirmatory clinical trials is because when you look at weak control, it's actually much different from strong control. It's actually a much weaker definition of error rate control. Should have been called extremely weak error rate control. And that is because you're looking at one specific configuration of parameters out of this boundless, unlimited space, unlimited parameter space. And because of that, it's actually possible, and we've published several review papers recently to give examples that you can actually cheat quite easily by injecting some tests that are known to have a large effect size, and then you can construct a really bizarre looking multiplicity strategy that becomes valid as soon as you switch from the strong control to the weak control. And that's simply because the weak control is not always the most meaningful, the most reasonable approach to defining error rate, the error rate. What I've done so far is I've defined the multiplicity problem, and let's now begin discussing solutions. And solutions may come in the form of gatekeeping procedures. And I get this question a lot. What exactly is a gatekeeping procedure? It's simply just an example of a multiple testing procedure which is built for a specific purpose. This is a procedure which is built to address multiplicity problems when the families are grouped into families. And there are two main requirements when it comes to valid gatekeeping procedures. The first one is obvious one. They need to provide some reasonable form of error rate control. For example, they should provide, I will assume in this presentation that we need to have global error rate control over all families in the strong sense. And if you think that this is actually difficult to achieve, it's not actually that high of a hurdle, because you can always apply a basic bound Fourier adjustment. You can control the alpha in any multiplicity problem, no matter how complicated it is. But this would not be an efficient solution. What we're looking for here is something that would actually account for the hierarchical structure of the problem. Once again, if you remember, we have several boxes of tests. We got more important, and then we have less important tests. That is something that needs to be accounted for. And this can be accomplished using the concept of optimal distribution of power. You would like to optimize power, statistical power, for the more important tests, and then whatever is left will be applied to the less important tests. And I will provide this slightly more formal definition of that. There's been a lot of research done in this area. In this slide, I would like to mention just only some of the approaches. This is obviously a very, very high-level list. There's been probably close to 50 papers published. It's been a very hot area of research. Instead of talking about those specific methods, I would like to emphasize the importance of defining certain principles that drive the process of building powerful and flexible solutions to complex multiplicity problems. And what I've got defined on the slide is, you can think of it as the ABCs of gatekeeping procedures, or maybe even multiple testing procedures in general. First of all, and this is requirement A, it's important to gather all relevant clinical information, which normally comes in the form of logical relationships or logical restrictions. B is we gather all available statistical information and try to get as much information as we get about the joint distribution of the test statistics or p-values equivalently. And finally, using that information, we define a number of candidate procedures. And then through the mechanism of statistical trial simulation, we choose the one that gives us high statistical power, and yet it is consistent with both logical relationships and available distributional information. So these are the general principles, and let's now, in the next few slides, I'm going to illustrate this general process by applying it to the case study based on the lurasidone phase 3 clinical trial. The clinical information that went into this process is summarized on the slide. The sponsor, realizing that endpoint E1 being the primary endpoint, obviously the very first objective is to demonstrate a beneficial effect on the primary endpoint, and only then to proceed to the secondary endpoint. From the sponsor's perspective in this case study, it was sufficient to establish efficacy at just one dose level, but highly desirable to get efficacy at both dose levels, and that's because it provides more information about the dose-response relationship, and it makes it easier for the prescribing physicians to choose the dose, for each individual patient, because there's more to choose from. And finally, which is commonly the case in CNS trials, there was no evidence of a positive dose-response relationship coming out of their phase 2 trials. So how do we incorporate this clinical information? This can be done by drawing connections. I showed you a similar diagram a few slides back, and that diagram only have 4 boxes. Now we begin utilizing the clinical information to connect those boxes to draw those logical relationships. From a clinical perspective, once we establish a significant effect on the primary endpoint at a certain dose, we should not just jump diagonally. We should always kind of work our way down and stay at that dose level. If we only win at the low dose, but not at the high dose, hypothetically, then the secondary endpoint at that high dose becomes, as a statistical test, it becomes more or less irrelevant. And that's why the very first thing that we do is we draw those vertical arrows. They really capture all of the relevant clinical information. And then the question, and this is something that we get a lot from clinical trial sponsors. How do we then use that clinical information? What if we just stick with something very, very simple, and let's now connect H3, which is the secondary test at the low dose, which H2, which is the primary test at the high dose. Why may this be attractive? Well, because it turns this complex multiplicity problem into just a simple sequential strategy. But if you think about this, actually there are lots of very good papers. For example, a paper by two FDA statisticians, James Hung and Sujane Wang. The simplicity is not really the goal here. Our objective here is to construct a testing strategy which is aligned with the clinical objectives. And from a clinical perspective, that this zigzag doesn't make much sense. What we should try to do is we should try to work from the primary family, the primary endpoint, down to the secondary endpoint. And therefore, now that we're done with the clinical information, let's now talk about how to best utilize the available statistical or distributional information. And this is where I begin introducing candidate gatekeeping strategies from which we will ultimately select the best statistical strategy. Probably the most basic approach here is to say, since we have two different branches, kind of vertical branches in this decision tree, let's take the overall alpha, which is 5% two-sided, and we'll just split it between the two branches. And we will apply, a simple alpha propagation rule, which means that if we are lucky, in this case the sponsor is lucky and they can reject H1, then the alpha level assigned to that hypothesis can be carried over to something else, for example, to H3. And once we achieve the bottom row in this matrix, whenever there's alpha which is released after a significant test, it can be carried over to another test within that family. It's a very reasonable strategy, and it has been applied to a large number of clinical trials. However, what I would like to point out here is that this strategy does not use available statistical information. This is a good test, but it's non-parametric, which means that it only uses the marginal p-values. We know that within each family, the test statistics are strongly statistically correlated, and that's simply because we have two doses compared to a common control. Since the comparisons share a common group, they will be correlated, and in a balanced design, the correlation is 0.5. Is there any way for us to utilize this information to our advantage and increase statistical power of this strategy? This can be done. In fact, we've built a general statistical framework that enables clinical trial sponsor to build those more powerful gatekeeping procedures. The basic idea is that you define those two families, and then within each family you apply a pre-specified powerful multiple test, and then whenever you reject something, then when you see a significant result, a certain fraction of the overall alpha is going to be released, and then you can apply it in the second family. This is it's obviously a very informal description. Formally, it's known as a set of alpha propagation rules. And as one example, and this actually goes back to a case study which was considered in Dr. Haddad's presentation earlier today. We can try to apply Hochberg type procedures within each family. The advantage here is that those procedures are more powerful. They're actually uniformly more powerful than the corresponding bound-for-any-based procedures, but they still control the overall alpha. They actually control the alpha within each family and also across the families. Let me make just one quick comment here. As you can see here in family 1, we're applying a truncated Hochberg test, and then in family 2 is the regular Hochberg test. Well, the reason we have to apply a truncated Hochberg test is because, as was explained in Dr. Haddad's presentation, and then Dr. Koenig actually gave a counterexample. We cannot use the full-blown Hagberg in the first family. It's a greedy procedure which is going to consume all of the alpha. Will be nothing left for the secondary family, and therefore what we do here is we apply kind of a general shrinkage type argument. We kind of shrink that test a little bit toward the Bonferroni procedure, and that results in a slightly less powerful but still very, very attractive Hagberg-based gatekeeping procedure. And now that we're done with the first two principles, now let's very quickly take a look at the very last step. Now that we have two gatekeeping candidate gatekeeping procedures, and normally we can have as many as 5 different ones that are aligned with the clinical objectives and also use all of the statistical study information. Let's now try to run some basic clinical trial simulations to see how those two procedures perform under plausible assumptions, and at the same time perform a little bit of sensitivity assessment. So once again, gatekeeping procedure 1 is based on Bonferroni derived tests, and gatekeeping procedure 2 is based on Hagberg type tests. Just as a quick example, here's a set of assumptions. Obviously, in real clinical trial applications, we do a lot more comprehensive assessment. Let's assume that each dose is powered at 80%. Each dose placebo test, is powered at 80% for the primary endpoint. This is the left-hand side of this diagram. On the right-hand side, we see family 2. That's our secondary endpoint, and we see here a slightly less power, 70%. When we summarize the performance of multiple testing procedures, I found it's very helpful to think in terms of multiplicity penalties, because if you think about this, any multiplicity adjustment is a penalty. So let's see how much penalty is actually introduced when we apply the first and the second gatekeeping procedure. We can see here that with the Bonferroni-based procedure, the probability, not actually the probability, but the multiplicity penalty, which is the difference between the original power and power after the multiplicity adjustment, expressed in in terms of percentage points, we're losing almost 9% of statistical power at each dose level. And if the sponsor is interested in demonstrating significance at both dose levels, then we're losing more than 11 percentage points in terms of power. With the on the right-hand side, with the Hagberg-based procedure, the results obviously look a lot more attractive. With each individual dose compared to placebo, the loss is only 4 percentage points. And when you look at the very last, at the rightmost column here, which is labeled both, which is again probability of finding significant effect at both dose levels. The beauty of Hagberg-based procedures is that they reward consistency. If the sponsor is truly interested in establishing an effect at both dose levels, you can see that the multiplicity penalty, instead of going up, actually goes down and drops to only 2.7%, which means that if it's highly desirable for the sponsor to win on both doses, that would be the way to go. Okay. And so what I discussed in the previous slide were the multiplicity penalties in family 1 for the primary endpoint. For the second endpoint, we can perform a similar assessment. We can see here that obviously the results in general look less impressive, and that's because the first family serves as a gatekeeper for the second family. We're going to lose some power simply because we have to go through that gatekeeper. But still, when we compare the Bonferroni-based procedure on the left-hand side and the Hagberg-based procedure on the right-hand side, it's clear, that there's a uniform improvement here. We do lose some power, but it's clearly more attractive to go with the Hagberg-based gatekeeping procedure. And once again, I would not suggest that Hagberg-based procedure should be used in all clinical trials. What I would like to, again, get across to use the message that it's very important to have a set of meaningful principles and then develop a systematic process of evaluating, based on clinical trial simulations, available options, and then choosing the one that is most robust and gives us highest statistical power. So in terms of evaluation criteria, we can actually summarize the results presented in the previous two slides using certain pre-specified criteria, using disjunctive or maybe sometimes conjunctive power or maybe weighted power. There are some papers that discuss the pros and cons of using those approaches. In this particular case, the sponsor in this Lurazidone phase 3 clinical trial, the sponsor decided to go actually with the Hochberg-based gatekeeping procedure, which I defined earlier in this presentation, and they were able to apply it quite successfully. All of the adjusted p-values, which is obviously a very important consideration in terms of regulatory review, all of them turned out to be significant, and therefore the sponsor actually got all of the labels that they were hoping to get out of this specific clinical trial. So I think I'm ready to close, and would like to emphasize once again that what I've discussed is a class of multiple testing procedures that enable clinical trial sponsors to control the global error rate across all of the primary as well as key secondary endpoints. They help sponsors maximize statistical power, and actually they've been widely used in clinical trial, in real clinical trials across the industry. I've given you just one example based on a real development program, and there are other examples in which similar principles have been successfully used. Okay, so I will stop at this point. Thank you so much.